Hello, everyone. My name is Ethan, and welcome to the Waterloo Nanotechnology Conference of 2020.
What just happened?
Clicking into it.
Stay on the line. Are the service okay?
Yeah, okay, let me check. I mean, everything looks fine. Oh my god!
What?
Batteries.
The battery's blown out.
The battery's blown out,
It needs to be replaced.
but the nearest store is 30 minutes away.
We could just make one.
Make one?
Yeah, I mean, you know, they're always in engineering. They're building stuff. I see them all the time walking around.
There are a million
Other ways to do this.
Fine, and you figure it out.
Ethan.
I swear to God, if you do this,
Yes!
What are the directions to the nanolab?
Yes, sir. So you're gonna run straight ahead, under the bridge, and then first door to your right.
Got it.
You got what I'm looking for. You got everything that I want.
You guys are so stupid.
No, you're stupid.
Once I got a piece of this ecstasy and heavenly bliss, I'm back for more.
Hey, sorry, can you hold this for a second?
Sure.
Thank you.
Now you're mine. Give me some more. Give me some more.
Hey, yo, stealing a bike? Nice.
this is illegal.
More of that, la 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 la. Give me some more of that, yeah. Give me some more.
Hey, what's the room?
It's room 101.
Got it.
It's 1102.
Wait, no. I'm here.
Yo, messed up. It's 1102.
Give me some more. Give me some more. What you got? Give me some more of that. La 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 la. Give me some more of that. Yeah. Give me some more. Give me some more. Give me some more of what you got.
Yes! Hey everyone, sorry for the technical difficulty. Hello? Hello?
Guys? All right. Well, good morning, everyone, for real this time. My name is Ethan, and I'm the chair of the organizing committee, and welcome to the 7th Annual Waterloo Nanotechnology Conference. I'm so glad all of you could join us here today. Now, this is our second year running virtually, and I'm sure we're all a little bit tired of sitting in front of our computers, but we really do have an exciting lineup for you today. This is my 4th year on the team, and every year we try to spice things up a little bit and run the event a little differently. So as you just saw, this year we decided to make an intro film, which was directed, filmed, and edited by our very own director of design, Mahmoud Haloum. So we hope you enjoyed it, and we will be posting it on our socials if you missed it or want to rewatch. Since we are running this event virtually, similar to last year, we want it to be a choose your own adventure. Since you're attending in the comfort of your own rooms, feel free to get up and walk around, grab a hot beverage, and enjoy. I know many of you came here for a specific reason, whether that be the energy storage panel or a keynote, but we're hoping that if you stay for just one more session, you might actually learn about something else that really piques your interest. And that's the magic of conferences. We've got a little bit of everything from biosensors to photonics, so we hope you enjoy your stay. Just some housekeeping items before I begin and introduce our first speaker. I have my team member, Caitlin here, who will give you and show you the ropes of Hopin. I know some of this virtual event stuff, some things are more intuitive than others, but we're just gonna give you a quick 1-minute rundown of how to use this, this platform so you can get up and running and don't have to worry about it for the rest of the day. So I'll hand it to you, Caitlin.
Hi everyone. So we're just gonna start on the left-hand side of the screen. You'll see the reception tab. So under here, that's where you'll find all your event details, including the schedule and all the speakers. Right below that is the stage, which hopefully you've found by now. That's where we're broadcasting right now. And then below that is the sessions. This is where you will find the breakout sessions today, and the panels and the keynote will be under the stage that we were just previously at. Next is the networking. So here you'll be able to start one-on-one conversations with anybody that's on the conference, and you can leave anytime. And then finally is our expos. Here you'll find all our sponsors. So if you're looking for a co-op or just curious about businesses in the field, you can find information here. We're also having research posters and presentations going throughout the conference. So drop by and check those out, and you'll have an opportunity to vote on which one you think is the best. On the right-hand side of the screen, there'll be chats, polls, and you can find questions or ask questions throughout the conference, and our team will be monitoring the chat. You can also, if you choose to schedule parts of the event, you can look in there. There will also be specific chats when you're in events such as the stage, and you can drop questions in there. And that's about it. So we hope you enjoy the conference, and let us know if you have any questions.
All right, perfect. Thanks so much, Caitlin. And one more thing before we get to the conference, this event certainly wouldn't be possible without our sponsors. So let me just pull them up right now. So I would like to take this moment to thank all of our internal sponsors at the university, the faculties of engineering and science, the Department of Nanotechnology Engineering, the Office of Research, and the Water Institute for Nanotechnology. And of course, our gold tier industry sponsors. I'd like to thank the team at Nerve and the folks over at Lumentum. You'll be able to find their booths in the expo. So if you'd like to talk to those teams, feel free to do so during the networking session. All right, so that pretty much wraps up all of our housekeeping items. Let's begin. So right now we're all tuning into the event through our electronic devices, our computers or phones, which are incredibly complex. And last year we discussed this at our conference. These devices only exist because of tools and techniques at the nanoscale, such as lithography. However, every day we carry around something much more complex, which is the human brain. In each cubic millimeter of your brain, there are hundreds of thousands of cells connected through billions of synapses. And so this is sort of the gold standard of complexity, and it would be a game changer if our devices, our phones, could replicate the performance and efficiency that our brains have. But to get to this point, we must first understand the brain, how it works. And who better to explain this than our next speaker? And our next speaker is Professor Edward Boyden. He's a professor of neurotechnology at MIT in the departments of brain and cognitive sciences, media arts and sciences, and biological engineering. At MIT, he leads the synthetic neurobiology group, which develops tools for analyzing and repairing complex systems such as the brain. He is certainly one of the foundational pillars in the neurotechnology community as a highly decorated researcher for his and his lab's contributions, but also a mentor in the field for his undergraduate and graduate students. And now, today, he's a teacher for all of us here at the conference. So it is my great pleasure to introduce our keynote speaker of the day, Professor Ed Boyden. All right, Ed, I'm just gonna add you to the stream now.
Hi, everybody. How are you doing? Can you all hear me and see the screen?
I can hear you just fine.
Great. All right. And is my slide sharing coming through?
Yes, now it is. Perfect.
Okay, thank you.
Good to go.
Great. Well, it's really wonderful to be here today, albeit virtually. Too bad we can't get some coffee in person and talk about the big ideas that I think will emerge in the time to come. I direct a group at MIT that works on technologies for studying and controlling complex biological systems, like the brain. And biology is fundamentally, I think, a nanoscale science. The building blocks of life are nanoscale, and they work with nanoscale precision. And so one of the big tensions is why can't we control, analyze, and map these things with the precision they need? But it's worse. There's also a time precision that we have to deal with as well. Biological systems are always evolving. And so one thing that we've been doing in our group has been trying to build technologies that really confront the spatial and temporal scales of biology head on. We want to see everything and control everything. Now, if we think about space and time very seriously, then technologies that are very different from what are out there start to suggest themselves. And so today I want to tell you 3 short stories where we took a way of doing things and came up with a very different way of doing it, sometimes the opposite of what people have been doing for hundreds of years, and how that's helping us understand and repair biological systems. So I'll tell you 1 story about space and 2 about time. And in all 3 cases, I hope you'll see why crossing these dimensions of space and time is so helpful if you want to see and control something as complex as a brain, for example. Let's start with space. Biomolecules are nanoscale in dimension, and they talk to each other with nanoscale precision. Why can't we see all of them? And this is a tough problem. Lots of pioneers have tried to solve it. In recent years, Nobel Prizes have gone to inventors of cryo-electron microscopy, of super-resolution microscopy, but no technique really lets you see everything in 3 dimensions with the nanoscale precision that we really want, especially if you want it to be easy to use, right? The technologies that have come out to date are expensive and slow. What if everybody could do imaging with nanoscale precision? And so in our group, we often think to do the opposite of what people are doing. And so we started wondering, for 300 years, people have used a lens to zoom in on something. What if we could actually just make the biological system physically bigger? We call this expansion microscopy. After many years of thinking about it, we started working on a strategy to create networks, spiderweb-like networks of swellable polymer, such as shown in the cartoon on the left, inside cells and outside cells, throughout biological systems, in between biomolecules, and around biomolecules. Do it just right and add water. The swellable material, which is a lot of the stuff in baby diapers, will absorb the water and make the biological system physically larger. So this, of course, builds upon some older strategies. In 1980, Toyoichi Tanaka showed that you could take highly charged hydrogels, like those made out of sodium polyacrylate, add water, and the highly charged network would repel and swell, as shown in the cartoon. Now, you can't just take the swellable polymer and dump it on top of the brain. You have to get it inside the brain cells, in between the biomolecules, around the biomolecules. In 1981, Christine Dreyer and Peter Hausen had a paper where they did just that. They used an uncharged hydrogel, polyacrylamide. We're using the charged hydrogel I mentioned on the previous slide, polyacrylate. But it's amazing how close they came to taking preserved embryos and tadpoles, synthesizing a mesh of charge neutral hydrogel, polyacrylamide, and at the time they showed that this could facilitate staining and imaging of these specimens. So our idea was to take a cell like the one on the left of this cartoon and to pull all the building blocks of life apart from each other. You end up with something like the thing on the right, a constellation of biomolecules hovering in space. Their relative organization is preserved, but it's physically larger. 2 biomolecules that were touching are now some distance apart, and 2 molecules that were some distance apart are now pulled apart to some macroscopic distance. Now, to make this work, we had to invent several additional chemistries, illustrated by the following cartoons. In this cartoon, proteins are shown in brown. We had to invent handles, little anchors, shown in purple. We now have created handles for proteins, DNA, and RNA, and are working on handles for sugars and lipids. Why the handles? Well, we have to give something for the polymer to pull on. We make the polymer using a recipe not unlike a mashup of the 1980 and 1981 papers I just mentioned. We had monomers, shown as little white spheres, and they self-assemble through a process called polymerization into these long chains. If a chain encounters a handle, the ones shown in purple, they form a bond. So if you think about it, the polymer can swell and the handles convey the force to the biomolecules. We're almost done, right? Well, there's one more step. The sample doesn't like being expanded, so we have to use chemicals, heat, detergents, enzymes to soften it up. And then when we add water, the baby diaper material will absorb the water. But this time, because of the handles and because of the softening, all the biomolecules will be pulled apart. Physical magnification of a biological specimen. We showed this in 2015. Fei Chen and Paul Tilburg, both students in my group at the time, were the co-first authors of the paper. In panel B is a small piece of the mouse brain. In panel C, the same piece of mouse brain about a day and a half later. The polymer starts at very dense. The spacing between polymer threads is only 1 to 2 nanometers, as shown in the sketch in the upper left. And after we expand, the spacing becomes bigger. Obvious question, is the expansion even? It's not perfectly even, but it's pretty darn good. We measured using a classical nanoimaging method, pre-expansion images, and then we would take the same sample and image it after expansion with a regular microscope. Here you can see in purple the pre-expansion image and in green the post-expansion image. The expansion is not perfect, but the distortion is really small, just a few percent over a typical microscope field of view. And that's acceptable for almost any biological or medical question currently being investigated. On the left, you can see a piece of the brain. Each white box zooms into the picture above. The left column is pre-expansion. The right column is post-expansion. The color code is shown on the left. We're using fluorescent antibodies to label, so they can be seen, different proteins. I hope you can see that the right-hand images are much more crisp than the left-hand images. In fact, the purplish blobs on the left become clean blue and pink regions on the right. We can see them more clearly because the expansion helps. The blue and pink areas are pre- and postsynaptic proteins. And if we look at the distance between them, we get the same answer that Catherine Dulac and Zhao Zhuang found many years ago using super-resolution microscopy, invented by Zhao. But this time, we can get this kind of information on a regular old microscope. Nothing fancy, just the same kind of microscope that people have at ordinary local facilities. So expansion really democratizes nanoimaging. Now anybody can do nanoimaging, which is in the end what we want, right? That gets down to the fundamental building blocks of life. In work we did with Eric Betzig's group, Ray Gao, Sho Asano, and Gokul Pariula showed that we could use antibodies against mitochondrial proteins, lysosomal proteins, shown in the upper right, myelin proteins, shown at the bottom. You can look at a wide variety of different molecules of great importance for biology. You can look at artificial proteins as well. Many fluorescent proteins have been made by mutating genes from jellyfish or coral. You can express these randomly and give the brain a color code. Every brain cell will look slightly different in color. Now, with a regular microscope, the fine wiring of the brain is too fine to be seen. Light has a finite size or wavelength, which means that very fine features are hard to see, as shown in the top middle. The top right, though, is the same region as the top middle, but after we expand. Color coding plus expansion lets you see the fine wiring of the brain. And many groups are now mapping the brain using these strategies. So far, what I showed you is all protein-based, but you can look at other biomolecules like nucleic acids. Here we're anchoring and expanding both proteins and nucleic acids. A fluorescent protein is shown in green, and the purple dots are individual expressed genes, individual messenger RNAs. So now, for the first time, anybody can map out different kinds of biomolecule using an ordinary microscope with nanoscale precision. By the way, you can introduce multiplexing as well. Many people have made libraries of probes that have barcodes on them that you can then read out through clever computational and imaging schemes. For example, we looked at dozens of genes that are cell type indicators of the cortex in the mouse brain. You can take these little padlock probes, shown in purple in the upper left, so-called because of their shape. Each one will bind to a different expressed RNA and have a barcode in it. You can then sequence the barcode. And sequencing is basically copying. You copy the gene over or the barcode by adding nucleotides that glow, and your microscope can read them out. By the way, this work and also work on the previous slide were done by Ozwasi, and also on this slide, Shaharlon, Dan Goodwin, Anu Sinha, and Fei Chen. Here we took a couple dozen genes found by the Allen Institute to be cell type indicators, clustered them in the upper left, and then used those to make padlock probes that bound to express genes across the mouse visual cortex. This is about 1 millimeter by 1 millimeter on the right-hand side. The color code on the right shows the layers of the cortex, and now you can see all the different cell types. In the upper left, you can see these kinds of clustering plots in many kinds of paper. And on the upper right, for the first time, we're able to see a lot of the cell types of the visual cortex with nanoscale precision in intact brain tissue. By the way, you can sequence more than just express genes. You can sequence the genome itself inside a cell. Andrew Payne, Zach Chang, and Paul Reginato, working with my group, Jason Van Roster's group, and Fei Chen's group, showed that we could knock little barcodes into the genome randomly and then sequence the barcode as well as the adjacent DNA of the genome. As you can see in the bottom, this allows us to not only get the shape, the sequence of the genome, but also its shape. How big can you expand? Well, everything I showed you so far is expanded by about 4 to 5 times. But you could take a polymer and expand something, form a second polymer in the space opened up by the first expansion, and expand it again. So if 4.5 times is 1 round, twice expansion gives you 4.5 times 4.5, or 20-fold expansion. J.B. Chang pioneered the first version of this when he was a postdoc in our group. Lately, Deblina Sarkar, Jinyun Kang, and Ozwasi, with great help from Margaret Schroeder, and Amuchi Abanari and Christina Kitko have developed important versions of this as well, we showed that we could do this with all off-the-shelf chemicals. Form a polymer, expand, form a second polymer, expand again. The cartoon at the top illustrates another advantage of doing this. Proteins and other biomolecules in a cell or tissue are all jam-packed together. You have to bring in labels, right, in order to make them visible. In the top half of the cartoon, the red and green antibodies are able to bind to proteins on the outside of a cluster, but they can't get inside the dense cluster. In the bottom half of the cartoon, when we pull the proteins apart, now the blue and yellow antibodies can also get in there. In other words, we can make invisible molecules into visible ones. We wondered, could we look at normal and pathological areas of the brain and see things that nobody had seen before? We decided to start by looking at synapses, the junctions between neurons that help them communicate. They are famously jam-packed with hundreds of different proteins. As you can see in panels B, C, and D, many invisible proteins turn into visible ones. Yellow is pre-expansion staining with fluorescent antibodies, and yes, there's not much yellow. And then purple is the same samples, same fields of view, same antibodies, but after we expand, there's a lot more purple than yellow. Not all proteins are as crowded, though. In F, G, and H, you can see that some proteins are not crowded, and the yellow and purple look more similar. In other words, we can now start to really map out many proteins that were not previously visualizable by any technology we know of. Working with Li Wei Sai's group on Alzheimer's disease, we wondered, in neuropathology, there are many proteins that are packed densely. Could they become more visible? Here we looked at Alzheimer's model mice. These are mice that have genes that make them get Alzheimer's-like symptoms. Again, yellow is pre-expansion staining, and purple is post-expansion staining. Same specimen, same antibodies. There's a lot more purple than yellow. In fact, if you look at the bottom, we even saw these purple dots that we never saw pre-expansion. Maybe why Alzheimer's is so hard to study and solve could be because we're looking in the wrong place for the most important, or at least some of them contributing, molecules that play a role. We saw the same dots with multiple different antibodies against the amyloid components, and we never saw these in wild-type animals. Here's another example where invisible things became visible. It's a crowded slide. This is work that we have now submitted for review, led by Pablo Valdez-Quevedo in our group. And that he is a neurosurgeon, and we studied brain cancer with him. We wondered, are we counting all the brain tumor cells? To make a long story short, if we labeled the brain tumor cells with antibodies after we expanded, we saw almost 10 times as many cells. These are actual human tissues from actual human patients. We saw almost 10 times as many tumor cells than if we stained before we expanded. That might explain, in part, why brain cancer is so devastating if indeed we are undercounting the tumor cells by nearly a factor of 10. How precisely can we expand? Well, when Ray Gao and Jay Yu were in the group, we wondered, Could you expand more precisely by using more modern polymers? As I mentioned, we use this 1980-1981 style of polymer synthesis, but many new polymers have been invented since, like this diamond lattice hydrogel that you see on the right-hand side of the slide in cartoon form. The hydrogels we used in the past have a lot of polymer defects, dense areas, dangling ends, and loops. These diamond lattice hydrogels, as shown in schematic form on the right, don't have those problems. We've synthesized these and decided to try to expand viruses, not just because they're interesting and important for human health, but also because they're sort of a natural scale bar. They help us see what we're doing at the nanoscale. This tetragel, as we call it, was able to expand herpes simplex viruses much more accurately than if we used the classical gel. The numbers are shown in panel G in the lower right. We were able to get down to single-digit nanometer imaging on a regular, ordinary microscope. This preserved the shape of the viruses much more accurately and might help us with analysis of viruses in the future. Of course, we can expand all sorts of different tissues. When Yongxin Zhao and Octavia Bucure were working with me, and Astrid Wines, Andy Beck, and many other pathologists, we showed we could expand a wide variety of human pathology samples. Prostate, lung, breast, pancreas. On the left are normal, and on the right are cancer-containing specimens. The color code is shown in the upper right. In all these different cases, from actual human patients, we were able to see more detail after expansion than before. So to summarize this first half of the talk, we have discovered that we can physically magnify systems in biology, cells, tissues, and organs. Almost 350 experimental papers and preprints have come out. It's a really easy procedure to do. Over 1,000 groups, I would estimate, are doing expansion experiments already. This slide just shows a snapshot of some of the experiments. People are looking at the human kidney, the microbiome, the fruit fly brain, plant seeds, bacteria. The list goes on and on and on. We think we have helped democratize nanoimaging, and anybody can learn to do it. Just visit our website, expansionmicroscopy.org. Pre-COVID, we had lots of hands-on workshops, but because of COVID, we had to shut those down. In the summer of 2020, we published a photographic step-by-step tutorial, which shows even very rudimentary skills, like how to handle a cover slip with a pair of forceps, or how to use a paintbrush to pick up a specimen. So many groups have used this photographic tutorial on our website to learn how to do expansion in the time since. Now, this is not a biology story, but we wondered, could we run expansion microscopy in reverse? Take a swollen hydrogel, laser print interesting materials inside, like quantum dots or DNA or interesting conductive materials, and then dehydrate it. If the expansion is nanoprecise, maybe the shrinking will be nanoprecise as well. This is work done by Dan Aran and Sam Rodikas when they were students in my group. In panel K, you can see a smiley face that we photo patterned, and on the right, when you shrink it down by 1,000 times or even 10,000 times in volume, we can make it much smaller. That means you can use ordinary lab equipment to make nanotechnology. Here in panel E, we wrote some lines of nanoparticles, again using a 2-photon laser to pattern these anchors and then attaching gold nanoparticles to them. We shrank it down, and then panels G and H, you can see we were able to make 50 nanometer wide lines, again with ordinary biology lab equipment. We were able to make conductive wires. Here we are, we patterned gold followed by silver intensification, and we were able to make low resistance silver wires using this photo printing and shrinking technique. So we're very excited now that the opposite of the expansion process, which we call implosion fabrication, could also be very cool. It allows anybody to potentially make nanotechnology. You can create interesting 3D structures, non-self-supporting structures, because there's a hydrogel holding things in place, all sorts of different materials. That concludes the first half of the talk, which is all about space. In summary, we can expand things and shrink things. Expanding lets us see nano features on ordinary lab equipment, and shrinking lets us make nano features using ordinary lab equipment. But there's an obvious limitation of all this if you're a biologist. These techniques will not work on living things. And so in the last half of the talk, I want to tell you about the observation and control of living systems. Let me take a sip of coffee here. And some water. The first story I want to tell you about is optogenetics. In optogenetics, we control brain activity using light. If you could turn a brain cell on, you could figure out what it would do. Does it trigger a behavior, a decision, an emotion, maybe even a therapeutic state. If you turn a brain cell off, you can figure out what it's needed for. You have to get light into the brain if you want to use light to turn the brain cell on and off, but you can do that by using optical fibers. So the big question is, if you want to use light to turn neurons on and off, how do you make the neurons sense light? Well, I became fascinated by a class of molecule known as the microbial opsin. These are 7 transmembrane proteins, such as those shown in the lower left. They exist in microbes found in, for example, salty water, such as shown in the upper left, and they convert light into electrical energy, for example. The first to be found was the light-driven proton pump. Archaea single-celled microbes use this to convert solar energy into chemical energy. People also found light-driven chloride pumps. These also respond to light and convert light into a chemical gradient. And finally, people found light-driven ion channels. These are found in single-celled green algae, for example. Flash forward to the current day. We've now found that all 3 of these classes of molecule have members that are safe enough, fast enough, and powerful enough to mediate the optical control of neural activity in the living brain. When Shirhan and Brian Chow worked with me, we showed that we could find light-driven proton pumps, express the gene in the brain using conventional gene therapy methods, shine green or yellow light on them, and you could turn brain cells off. In the middle column, Xue and later Amy Chuang found that certain light-driven chloride pumps, if we express them in the brain, would allow us to turn brain cells off using yellow or even red light. Red light is helpful because red light goes deeper in the brain than other colors of visible light. Finally, light-driven ion channels we found can be used, for example, to turn brain cells on. Carl Dijsselroth and I collaborated on the first study on this in 2005. We expressed a channel rhodopsin, or light-driven ion channel, in neurons and showed that blue light could activate them. Later, Nathan Klepecki in our group showed that we could get a red light activated molecule, again helpful because red light goes deeper in the body than other colors of visible light. In recent years, we've been trying to push these to their physical limits. I mentioned that Amy Chuang found a red light-driven silencer. We could turn brain cells off with red light if we express this molecule, which we call JAWS, in the brain using a gene therapy vector. Nathan, when he was in the group, found crimson, a red light activator of neurons. Thousands of people are now using these molecules to turn neurons on or off and figure out how they trigger different pathologies or different healthy states in model organisms such as mice. Neuroscientists use this all across the board. But in the last couple of years, a very interesting second possibility has emerged. Could you use these molecules in people to help them? Recently, a group in Europe announced that they took our molecule, crimson, and delivered it using a gene therapy vector to the eye of an actual human patient with blindness. He no longer could sense light. But our light sensor, delivered to spared cells of his eye, allowed him to have partial restoration of functional vision. He could see the lines of a crosswalk and pick up household objects. Other molecules that we have might also have other potential. We found a very fast molecule called Chronos, for example, that might help with the restoration of hearing, and so on and so forth. Others have used the tools to make interesting discoveries. For example, Li Wei Sai's group, in work led by Hana Iakurino and Annabelle Singer, used this light-sensitive strategy, which we call optogenetics, opto for light, and genetics because we deliver genes to the brain, to discover that certain brain waves might help with Alzheimer's disease treatment. They delivered the optogenetic molecule that allows us to do blue light activation to the brain of an Alzheimer's model mouse and showed that if you drive the brain 40 times a second, the Alzheimer's got cleaned up. They later showed that you could deliver the same pattern with a 40 hertz movie. In other words, having the animal watch a flickering light was able to induce the same brain wave without the need for the optogenetics, no need for an optical fiber going into the brain. And now there's growing excitement about the possibility of delivering flickering lights and now clicking sounds as well in order to help in actual human patients to reduce the burden of Alzheimer's disease, one of the most intractable diseases of our time. So optogenetics, to summarize, is in widespread use. It's being used in humans now to explore as a treatment for blindness, and it's been used to derive new potential therapies for diseases like Alzheimer's disease. But I just want to emphasize how lucky we were. This was serendipity. These molecules didn't have to be safe or effective or fast. They could have just been toxic when we put it into the brain. And so that leads me to the final story I want to tell you today, which is our attempts to develop the opposite of optogenetics. It's great that we can control brain cell activity, but can we watch what brain cells are doing as well? Well, in this case, the natural world has not been so generous to us. For optogenetics, the natural world gave us this gift. It evolved molecules that let us control the brain with light. For observing things, not so lucky. So Erica Young and Kirill Pyakovich, when they were postdocs in my group, we decided to make our own luck. We built a robot that does evolution in the lab. After all, if the world won't evolve as something, maybe we can. Directed evolution has a long and storied history, but we decided to do it in mammalian cells because we want tools that work in the brain. And we also did what we call a multidimensional screen. We want molecules that are fast and safe and effective, not just one of the three. And evolution only gives you what you're screened for. Here's the schematic of how it works. We can take a gene and make millions of mutants. For any given goal, some are better and some are worse. Put them into cultured mammalian cells and scan them with a microscope. Some of the cells will have what we want, and we can then use a robotic arm to pull out the cells and therefore the mutants that we want. We did what might have then been, might still be, the largest mammalian cell-based directed evolution screen. We wanted to build a fluorescent voltage indicator, the opposite of optogenetics, if you will. It would light up when a brain cell was active. In 2 rounds of directed evolution, we screened 10 million mutants of a gene called Quasar2, found by Adam Cohen's group at Harvard. And we ended up with a molecule that worked really well, which we call Archon. This is work that we did in collaboration with Shuo Han's group at BU. We expressed Archon in the brain of a living mouse, and then using a simple microscope like shown in the upper left, shine red light and collect the infrared light on a camera, we got the traces you see on this slide. They look like electrical traces, but they're not. These are brain cells that are lighting up when they're active. So now we're very excited. There's so many biological processes we wish we could see. Can we make new indicators of all of them? Our robot might be able to help. Now, we're not the first to try to make fluorescent indicators of living functions. This is a screenshot from Jin Zhang's database from her group at UC San Diego. Hundreds of fluorescent biosensors have been made. I think the main role of our robot will be to help improve them by allowing multidimensional screens in mammalian cells. These fluorescent biosensors are often a fusion between a glowing protein, like one from jellyfish, and some kind of binding domain that'll bind to a biomolecule like cyclic AMP or calcium. Now, cells are really complicated. Wouldn't it be great to see how all these different biomolecules interact? Ideally, you could image many things at once in a living cell. That's really hard, though. Shannon Johnson and Chengyang Lingu in my group have been working on a new way to do this, though, that might be very simple and powerful. What if we could take 2 different fluorescent reporters, like the kind I showed you in the previous slide, put one of them at dots in a cell labeled 1, and put the other one at dots in a cell labeled 2. In other words, park them at different places in a living cell. While the cell's alive, you'll get a little movie from each dot of what's going on with that signal. Maybe the dots labeled 1 will tell you about the cyclic AMP there. Maybe the dots labeled 2 will tell you the calcium there. Later, you can go in using techniques like I told you in the first half of the talk and stain the cell and figure out which sensor was at each dot. Here's one way we can make these little nanostructures. We can take different fluorescent reporters and fuse them to different self-assembling peptides. Then they'll cluster at different, non-overlapping, stable spots within the cell. Later, you can come in and stain with an antibody that binds an epitope that distinguishes each reporter. That tells you which reporter was found at each dot. Here's a little animation made by the Ella Maru studio for us that illustrates the idea in a little movie. While the cell's alive, you see a bunch of green dots blinking at you. When the cell is done with its experiment, you can then take it and preserve it and stain it. The false color image would then tell you what indicator was at each spot. Amazingly, it works. Panel B is a much-loved calcium indicator called GCAMP6. Panel C, we took GCAMP6 and fused it to self-assembling peptides in a cultured neuron, and we found that indeed it will form stable, safe clusters at different points in the cell. We did lots of experiments and didn't see any changes in cell health, physiological dynamics, or sensor performance. So clustering the indicator does not hurt the cell. The moment of truth. We took 3 different indicators and connected them to 3 different self-assembling peptide sets. The green images show the cultured neuron while the cell is alive. We see a bunch of green dots with different indicators. While the cell's alive, they'll blink out little movies of what's going on there. When we're done with the experiment, we preserve it and then stained with fluorescent antibodies that bind the epitopes that tell the different indicators apart. That's what you see in these traces. We've done the movies with our microscope, and then we've then reconstructed which dot is at each point by using the staining. In this case, we gave a drug called forskolin, and we see all sorts of signals change, including cyclic AMP, calcium, and the downstream molecule protein kinase A. This is very powerful, right? Now you can look at how different signals within the same cell relate to each other. I'll just give you one example of the kind of relationship you can now see that was difficult and sometimes even impossible to see before. In panel H, some neurons, when you give them forskolin, have slow calcium. In panel I, some neurons, when you give them forskolin, have fast calcium. We wondered, does the speed of the calcium relate to the output, the protein kinase A? Well, in panel L, you can see the answer is yes. The fast calcium neurons had strong protein kinase A, and the slow calcium neurons had weak protein kinase A. So now we can derive relationships between different signals within the same cell. Hopefully we can figure out how cells compute outputs from their inputs. You can add a fourth indicator. Here we added a protein kinase C indicator. Or you can even add a fifth indicator. Here we added an ERK kinase indicator. So we're very excited that we can start to take a lot of these off-the-shelf indicators that already exist and use them in novel combinations to map out the computations of cells. To conclude this last technology part of the talk, we found that although we were not so lucky with imaging brain cells as we were with optogenetics, we can do clever tricks to make it more easy. We can build robots that evolve molecules that do what we want, and we can use molecules in clever ways, for example, putting them in different parts of a cell so we can see how those signals relate to each other. Going forward, one of my dreams is to combine the tools. What if we could watch all the activity in a whole brain, control it with optogenetics, and make a map with expansion of all the building blocks of life? Could we make a model of the brain and how it computes? Well, we're starting to try to walk down this path by looking at small brains, like fish and worms. The story of biology tells us, solve something small completely. After all, the first genome to be sequenced was not the human genome, it was a small virus, but that opened up the field. When Limor Freifel was in the group, she showed that we could do expansion microscopy of the larval zebrafish brain with only 100,000 brain cells. J.U. did something similar for the worm C. elegans with only 302 neurons. So we are very confident now that we can start to make maps of these small brains in their entirety. Erica and Carol, when they were in the group, showed that we could do voltage imaging in the zebrafish brain and in C. elegans, the worm. So we're very excited now to integrate these tools and to help understand how brains compute things like decisions and emotions. I'll end by just pointing out that we share all of our tools as freely as possible. Please visit our website, synthneuro.org. Don't hesitate to email me. My email is just my name, edboyden@mit.edu. And this slide acknowledges all the people in the group and our alumni at the top who helped with the projects. I think I've acknowledged all the people who led the projects along the way. In the middle of the slide is an even longer list of collaborators all over the world who've helped us develop these tools and to apply them. And so with that, I'm happy to take questions if you like. And yeah, stay in touch. We're always happy to help solve major problems with new technologies.
Hi, Professor. Thank you so much for sharing. I really enjoyed your talk. We have a few questions for you. The first one that I wanted to ask from the audience was, what challenges have you had to overcome in your research? Since your work has many applications, what do you find most rewarding about your work?
There have been lots of challenges.
Imagine.
Yeah. Well, yeah. I mean, we really are trying to pave a technology path into the unknown. Maybe I'll try to answer both questions at once. For me, I've always been very interested in understanding and improving the human condition through science. It's a philosophical quest for me. You know, when I was a child, I really wanted to know more about what it means to be human. Why do we have thoughts and feelings? Can we achieve a more enlightened state? And so I guess I've always kind of pushed myself really hard to try to be on the edge of what is possible. You know, I started college relatively young. I left when I was 14 years old to go to college. I tried to start what I think might have been at the time the first group solely devoted to tools for mapping and controlling the brain. That was a challenge. When I wanted to get a faculty job and become a professor, I was rejected at many of the places I applied to. That's how I ended up as a professor in the MIT School of Architecture, of all places, before finally moving into the neuroscience department full-time a couple of years ago. Yeah, it is difficult. I think there's a trick where you do want to push the boundaries of reality in the positive direction, but you still have to stay in the game. And again, I'm a big believer in luck and serendipity. The great discoveries are often driven by chance. And so it's also very tricky, I think, to be lucky a lot. But I try to teach that as a skill set in my group. I do think that luck can be taught because, there are ways, for example, to, as I mentioned a couple of times during my talk, we read a lot of old papers in my group. And, if there's 1,000 people working on something, I don't feel the need to work on that. But if you look at old papers that are now forgotten, there's lots of treasure hiding, and you can be lucky by looking for things that other people have forgotten.
Yeah, awesome. Was there any one specific moment that you think was maybe the highlight of your research so far?
Oh, there's so many. I mean, it's so fun to see something for the first time. When I did the first light activated neuron experiment at 1 o'clock in the morning in a dark lab, fully abandoned, that was so much fun. Seeing an expanded brain that was 100 times bigger in volume than it should be, that was kind of surreal. I feel very lucky to have been able to see lots of things, right as they're happening, and that's one reason why, as somebody once said, the best way to predict the future is to invent it.
Yeah, that's definitely one thing I found very interesting about your talk was hearing about being able to use just ordinary equipment for seeing nano and making different nano applications. That's something that there's another question about is why do you think that your expansion techniques aren't super widespread, and what do you think is holding it back?
The expansion techniques are actually spreading pretty quickly.
Oh,
Almost 350 expansion experiments and preprints are out there. I would
mhm.
estimate that about 1,000 groups are practicing it. I think it's spreading very quickly. People are applying it to everything from, trying to do early detection of cancer, to analyzing, the gut microbiome, to looking at how nephrotic kidney disease looks. If it's not widespread in your corner of the globe, drop me a line. We're very happy to share all the tools. You go to expansionmicroscopy.org. Anybody can download the papers and read how to do it. Yeah, it's spreading very, very quickly, which is exciting.
Awesome.
And
Well,
because it is so easy to use, right? I'm a big believer in democratization of technology and making things widely available, and optogenetics spread, quickly as well. I think because anybody could do it.
awesome. And then one more question. Are there any limitations to the expansion technique itself? Can the molecules break or provide unsaturated resolution images when the expansions are added upon each other?
So when we do expand the sample, we are treating it with chemicals and heat and other things. And so proteins we can expand away from each other quite well. We're still working on ways to do expansion of proteins and RNA away from each other so that we can do post-expansion staining of each. That's still a work in progress. But there are different protocols that can specialize in different priorities. So if it's not clear from the website what's best for your application, don't hesitate to email me. We help lots of people get going on expansion. And now, especially that people are seeing the dramatic improvements that occur with expansion, such as finding out that the plaques that occur in neurodegeneration are not where you thought they were, or that people are undercounting brain cancer cells by almost a factor of 10. And I think as more and more groups are realizing the dramatic improvements you get with expansion, I think more and more people will be applying it in different subdomains of biology that traditionally have not used, well, in many cases, have not used microscopy much at all. But have been more biochemical in nature. Yeah, so if anything needs any advice or tech support, drop me a line. Always happy to help.
Awesome. Thank you so much for joining us. I found your talk so engaging, especially with the cartoons that you had in the graphics. That was so useful for me to kind of connect what you were talking about to a visual. So thank you so much for that. Yeah, those are all the questions we have for you. If you have anything else to add.
Yeah, well, I guess number one, yeah, so we do take democratization of technology seriously. So if you need any help or want to use any of our tools, don't hesitate to drop me a line. And number two, again, I always believe in serendipity, and that we have to if these problems that are really, really hard could be solved by somebody alone thinking in their basement, it would have been solved by now. So if there are interesting ways to connect the dots and solve big problems, also drop me a line.
Awesome. Thank you so much.
Have a
Well,
great
yeah, we hope that you stick around throughout the conference. Feel free to jump through if you have time.
Great.
I'm
Thank you.
sure people would love to talk to you.
Wonderful. Yeah.
Yeah, so next we're going to be shifting to the energy storage panel. So that will be coming to you. Oh, just kidding. There's another question for you. What's the first paper of his that he recommended? Oh, do you have that by chance?
What was the question? Sorry.
The first paper that you recommended?
The first paper I recommended for what purpose?
Um, you know what? I think that we will determine that after. We'll clarify the question and get back to you if we have.
Great. All right. Yep. If anybody has further questions, just email me at boyden@mit.edu. Always happy to chat.
Awesome. Well, thank you so much.
Have a great
Thank you. So the energy storage panel will be happening next. Joining us will be Nian Liu, an assistant professor at the School of Chemical and Biomolecular Engineering at Georgia Tech. John Philippe Lee, an alumni of UW's Nanotechnology Engineering program, who has been working in battery R&D for since 2017. And Shiva Adireddy, the chief executive officer and co-founder of Advano, working to accelerate commercialization of battery-grade silicone material from lab to market. So stay tuned for that. As soon as our panel is ready to go, they will start screen sharing. So